How intelligent am I or you are? Where does our intelligence come from? Does it come from mother or does it come from father? Is intelligence inherited? Is a question. And often we would say it as intelligent people will get intelligent children. And the first point, of course, is to know the definition of intelligence. It itself is very difficult to describe. Nevertheless, people have been trying to find out several ways of what is intelligence or how the inheritance of intelligence happened. It is multifactorial. There are several reviews you can try to go and read about it, the new genetics of intelligence. And mostly what is found is that some of it is based on our DNA, that is, the genes determine some part of our intelligence, and then environment or how the training and the orientation, drive, motivation are influence the thoughts and focus of the individual would eventually translate into IQ or intelligence in general. So basically it's like the DNA or the DNA that we were discussing will create a brain, a structure, and the brain is influenced by the whole world around, what is perceived, absorbed, and also the amount of time that is there, and many other things, and that will influence the social influences, environmental influences, and the needs and the pressure and stress could also influence the way the brain develops, and therefore will also have effect on the intelligence. And I hope we all recognize that there are multiple intelligences, that it depends on how the output is. Somebody is good at singing, dancing, or somebody is at mathematics, somebody at imagination, poetry, many other things. So we will discuss about inheritance, and we will start with the very simplest way that we are ought to discuss. I am Dr. B.C.M.R., Assistant Professor at Shastra. We will talk about Mendel's laws of inheritance, and that is how that could be applied to various fields, and studying it at the fundamental level is an important thing for us. Before we start on the laws of Mendel's or laws of inheritance, it will be quite imperative that we learn the terminology. Biology is 50% memory, and memory of it majority of it is about the jargon, the names, terms, and many other things, processes. So here I think before we go ahead, it's important to just for those who are studying it for the first time, it will be important to understand these meanings of these. Phenotype is anything that is observed or which is a product of something else. A trait, for example, is a phenotype. We can look at it, we can see the effects of it, and that is a trait. Genotype, on the other hand, is the responsible gene, the gene responsible for that trait. So that is phenotype and genotype. Say for example, our skin color is a phenotype. Intelligence could be a phenotype. Our height could be a trait as well. Many things could be, all of these observed things could be traits. And the corresponding genes or respective genes responsible for these traits would have been the corresponding genotype. So this is about the genetic information that dictates the output or outcome, and that is the trait or phenotype. Then we will also discuss about haploid and diploid. We are humans, and we have about 23 chromosomes, 23 pairs of chromosomes. Say chromosome 1, we have 2 copies of them in majority of the cells. If we look into our body and or any other majority of the organisms, we have 2 parts. One of it is the somatoplasm and the other one is the germplasm. Germplasm is responsible for giving rise to all the gametes in our body that will be used for procreation for the next generation. Somatoplasm is all the body that we have as a result of fertilization that occurred from the gametes from our parents. So somatoplasm is diploid in the sense it has 23 pairs, that is 46 chromosomes, and germplasm usually is haploid. That means each of the sperm or egg, if we take mature gametes, they would have 23 chromosomes, not 23 pairs of chromosomes. So when we see that there are 23 pairs, we would also denote it as 2n, n representing the haploid set, and in the germplasm we have it n. N is haploid and twice that of it is the diploid. So if we look into majority of the organisms, excluding some bacteria, viruses, and some cases, we have a cycle of being diploid and haploid. Say diploid organisms will produce haploid gametes, and there would have been another diploid, say a male and a female, and it would produce gametes as well. These 2 gametes will mix together and produce a diploid organism, which will keep on will produce haploids gametes, and then the cycle keeps on going. And now there are important questions about what is a gene. Gene is, as such, to describe is difficult, but we will get to a working definition where gene is a sequence of DNA, or it could be RNA. It's a sequence of nucleotides that produces either RNA, or from RNA it could produce a protein. Right? So a gene is a nucleotide sequence which is transcribed to RNA, and by itself could be functional sometimes. Like we have genes for rRNA. rRNA is a non-coding RNA that it is a part of the ribosome, and its function is to act as a part of the ribosome, and so those are also genes. In some cases, or majority of the cases, the RNA is further translated by ribosome to produce proteins, and the protein has a function. So those sequences that have those sequences of the genome usually is DNA. Those sequences of the genome that produce functional RNA or functional proteins are referred to as gene. It is a gene. So if we take a gene X, gene for some property or trait or phenotype, there could be varieties of it. Say for example, we have a gene here. So this is the direction to be read. As we progress, we will get used to the way it is described. A broken arrow here represents the promoter, and this would have been the start codon. Upon transcription, mRNA is produced, and that will give rise to a protein. The protein is now functional, and it would have given an enzyme or structural protein it could be. So in every genome, we can expect that there are some changes. That means there are different forms of gene X. Right? Mutations keep happening, or there could be insertion deletions, which are also other kinds of mutations. So gene X, which was originally the most functional one, or the normal one, we can call it as the wild type, for example, it may have alternate forms, like say for example X1, or we'll call it as X2 because there is an alternate form. We'll call this as X1 and X2. There could be gene X, an alternate form for gene X as well. All these are referred to as alleles, right? So gene is something that codes for a functional RNA or protein, and alleles are different forms of that particular gene. Say here X1, X2, X3 are alleles of gene X. Okay. So we will also talk about a little bit about dominant and recessive. They are also referred to as dominant, law of dominance, and so on. Say, for example, there are some alleles. X1, say for example, will produce a protein, and X2 allele produces a non-functional protein, right? So here is X1 produces a functional enzyme or protein, and X2 produces a non-functional protein. So if we recollect, X1 and X2 are alleles. Their phenotype, in this case, because as we are discussing or putting it that way, X1 produces a functional protein, whereas X2 produces a non-functional protein. So in that case, we would observe a phenotype in this case, whereas for the X2 allele, there is no phenotype, right? Because it's a non-functional enzyme. So this is just for understanding sake. We have different variations to it as well. So basically, if we have a diploid, if there is an organism that is diploid, that means we have two copies of X. If X1 and X1 are present, then we call it as homozygous. Homo is same. If an organism has X1, say X2 and X2, then it is homozygous for allele X2. If an organism has X1 and X2, then it is heterozygous, hetero for different. So we can think of dominance and recessive in this case as well. Say, for simplicity, we all, as we discussed here in this case, that X1 and X1 produces a functional protein, whereas X2 produces a non-functional protein. Therefore, X1 will produce a phenotype, whereas X2 will not have a phenotype. So if an organism is homozygous for X1, something like that, then it is having the production of a functional protein, therefore the phenotype is present, right? And if it is X2, there is no phenotype here, because both X2s are producing a non-functional protein. Whereas X1 and X2, if it is present, that is heterozygous for X1 and X2, then what would you expect? Would you expect a phenotype or would you not expect a phenotype? Because X1 is producing a functional protein, there is a functional protein produced, and one allele is producing a non-functional protein. So the phenotype would be visible. And that is simply, or at the molecular explanation of, or genetic explanation of what is meant by dominant and recessive. Here, allele X1 is dominant over allele X2. Okay, that's how we can simply put, which means that presence of even one X1 allele will exhibit a phenotype. That is dominance. In this case, if we see that even though there is a there is one X1 and one X2. There is a phenotype, and that is because of the presence of X1. X1 is dominant over X2. If a recessive allele should be there, if a recessive for a phenotype for recessive allele should be there, then it should have been in homozygous. That is only when a recessive one can be, the trait for a recessive allele can be seen, right? So that is dominant versus recessive. I think we already covered the homozygous and heterozygous. Here we see X1, that is homozygous, and we have here heterozygous. So we will discuss about monohybrid and dihybrid in a later video, but simply put, when you say hybrid, if we have X1 like this, that is kind of both are same, so it's not a hybrid. And if we are basically, say for example X2 and X2 also, and during gametogenesis we will have X1 in different gametes, and X2 and X2, they are haploid, and upon fertilization it would form X1 X2, right? This is a hybrid in the sense we are talking about one phenotype or genotype, that is about X, gene X, or alleles of gene X, and we are talking about hybrids that are forming between two different parents. So we are talking about one allele, so we call it as monohybrid. One trait, so we call it as monohybrid. If we were discussing about two hybrids, two alleles, or sorry, two phenotypes, then we would be calling it as dihybrid cross. We will discuss this again in the later videos. I hope you got the idea of what do these mean. It's always in biology especially, there are too many words, too many processes, technical terms and phrases which we need to mug up many a times, but it's also simpler if we try to understand, try to associate break up the word and try to remember it for a longer time. Then it will become easy to read the textbook as well as research articles in the future. Thank you. Have fun.